Continuing with the RNA, I have given you in the previous class about the m-RNA structure, right? So m-RNA is messenger RNA that is formed by the DNA during protein synthesis while transcribing the messages. So what is transcription? I told you it is transfer of or copying of nitrogen-based messages from one part of the one strand of DNA which is acting as a template to m-RNA. That is called transcription. But you must be knowing that all the RNA are produced by the DNA itself. They are not produced on their own. Therefore, second RNA what I am explaining is about the t-RNA or the transfer RNA. So I am giving you now the structure of t-RNA, transfer RNA. So what is t-RNA? I told you its name is transfer RNA. What is that meaning? Transfer RNA is otherwise called as adopter RNA. So why the meaning adopter came? Because you should know, or you already know, that in protein synthesis during translation the messages of the mRNA are read 3 at a time as a coding languages, triplet codon language, and be translated to amino acid language by transfer RNA. Then transfer RNA will get the particular amino acid based on the codon with its anticode and get the mRNA by binding with the mRNA, by binding with the amino acids forming amino acyl tRNA and bring it to the ribosomes for translation. Therefore, this is adopting the amino acids for a shorter time. Therefore, we call it as adopter RNA. Right? Then the structure of tRNA was given by scientist Robert Hawley. Robert Hawley, you have heard his name, I believe. He was the one who did immense work on the DNA and RNA and even genetic code, and he was awarded with the Nobel Prize along with Marshall Nirenberg, Hergovin, Trna. Right? So Robert Hawley gave the physical structure of tRNA, which looks like a clover leaf model. It's called a clover leaf model of tRNA. It looks like a clover leaf. What is the meaning of clover leaf? Clover is the leaf which is having trifoliate character, and it will be there on your cards. Cards will be clover shape, right? You would have heard clover will be like this, right? I think you have seen this also. This is clover. It looks exactly like that. tRNA looks like clover leaf. Therefore, it is called as clover leaf like model or clover leaf model of tRNA. So how it appears to be? Whenever you observe, it is again a single stranded structure, but it forms number of loops in between because there is a false base pairing among some of the strands. Some of the nitrogen bases are falsely base paired, and those who are unpaired remains like loops. Right? So just imagine, suppose this is a RNA 5' to 3'. Imagine there is something like A here, and suppose here it is C. And here you have U. You know that A and U are complementary bases. So therefore it may undergo pairing like this. That is A pairs with U, whereas C will form loops. Here all A will be there, and U will be there, and C form the loop. These are loops are formed in the structure of tRNA. Therefore, such structures in tRNA I show 3 types of loops formed. Right? So how the diagram or how the tRNA looks like. So first I am showing you the strand which is called the main strand of tRNA, which will be having a structure like this. This is the main strand of tRNA, and it will lead to side strands leading to the formation of loops. This is one loop. As you go down, there is another loop, and the other side another loop, and these loops will have one strand which is an extra output. This is the structure. General structure, you have to draw 3 loops like this, right? So loops you can still enlarge them also. They are bigger ones also, right? So let us make double complete structure, thick structure of this tRNA. You are supposed to draw this. for 2 mark or 5 mark question. Okay, this is the tRNA. This is one loop at the left side, one at the right side, and one of the strand is straight and it is more longer than the other strand. And this is the 3' OH end. This is the 5' end. Okay, this is the structure of tRNA. Here you get the base pairs, which are actually false base pairing happening in the tRNA. This is again false base pairing. So these are the false base pairs. Means they are not actually present like a DNA, but they are false base pairs. And here are the unpaired bases forming the loops. Unpaired bases, all these are the unpaired bases forming the loops. Okay. Now, what happens exactly? The 3' end amino acid is brought. Therefore, we call it as the amino acid acceptor site or acceptor site. Okay, it will accept amino acids forming amino acid tRNA. And this first loop is called as a DHU loop or dihydroxyuridine loop. Next loop is called as T-C loop. That's the second loop. And the lower one is called as anticodon loop. Okay? Because here at the tip you get 3 anticodons which are complementary to the codon messages of mRNA. Maybe CAG is the anticodon. This is the anticodon. Okay? And this we call it as a variable arm. This is the total structure of tRNA. You have to draw and explain the parts of this. Now let us understand what are their functions. This part is called amino acceptor site, and this portion DHU is involved in the recognition of amino acids. So let me write here the functions. 3' OH is acceptor site and DHU refers to dihydroxyuridine loop. Uridine loop. Okay. This helps in identification of amino acids are called as amino acid recognition site. Right? This is the first one. Then second is called T-C loop. This helps in recognizing the ribosomes. It is the ribosomal recognition site. Okay. Then third one is anticodon loop. Anticodon loop contains 3 anticodons that are opposite or complementary to the codon messages. So this diagram and its explanation will be asked. First thing is how it looks like. It looks like a clover leaf-like structure, and it was given by scientist Robert Hawley. Then, according to him, it is showing a clover leaf-like structure. Clover leaf refers to the structure having 3 loop-like structures. Clover shape. It looks like a clover shape. Then, how are these formed? The loops are formed. Let us now enter into the structure. According to Robert Hawley, it is the structure of 3-looped structure, clover leaf-like structure. It's actually 5' to 3' is single-stranded RNA only, but here and there it will undergo. See, this is called stem of tRNA, where it has undergone false base pairing. This part we call it as the stem of tRNA. Stem is the strand. Stem of tRNA. Okay. Then the loops. DHU loop is the one, dihydroxyuridine loop I have written here. Then T-C loop, the other one, which helps in identification of ribosomes. The anticodon loop helps in identifying, or it has the 3 codons which are complementary to the codon messages of mRNA. 3 nitrogen bases of mRNA form one code, and that code will be reverse or almost complementary. It's complementary to the anticodons. Based on this anticodon, it will get the amino acid at the 3' OH end, and at this area you get CCA as free nitrogen bases. These are not going to bind with the other one. The upper 3 nitrogen bases are free, that is CCA nitrogen bases. This structure you have to draw, label and understand. And this is a 2 mark or 3 mark question, and for the board even the other things about the Robert Hawley and the clover leaf structure, why it is called adapter RNA, that and all is a neat part of it. Okay, now you draw this diagram and label it and write a short note on t-RNA. Okay, that will be your homework. So after writing t-RNA, you will have to understand how it is formed, when it is formed. It is formed by the DNA. Normally the DNA part, one part will be forming the tRNAs, and rRNA is the third type, that also I will give you. But tRNA is actually short-lived RNA. m-RNA lives for 3 to 5 minutes, but tRNA is still short-lived tRNA. Okay, done then. Coming to the other types of RNAs. So one already we discussed, that is m-RNA. Second one we discussed, that is tRNA. Then the third type is called rRNA. rRNA refers to ribosomal RNA. Ribosomal RNA. Why it is called ribosomal RNA? Because it forms the major internal part of the complete ribosome is formed by the rRNA. Therefore, ribosomal RNA is the normal one, which is simple thread-like structure but highly coiled, present within the smaller and the larger subunit of the ribosome. Like this, rRNA will be present. Then rRNA forms totally around 80% of total RNA. Means maximum RNA found in the cell is only rRNA or the ribosomal RNA. And ribosomal RNA helps, suppose this small subunit, the larger subunit, it will be like this, and mRNA binds to this. So mRNA binding will be made firm by the rRNA on the ribosomes. Therefore, major function of rRNA is to produce the ribosomes, main structure of ribosome, and also it will help in binding of mRNA to the ribosomes. Okay, that's about the rRNA. So totally 3 RNAs, general structures and their explanation, their characters, their structures were learned. rRNA, we don't have specific structure, simply it is arrangement of linear sequence of nitrogen bases. Okay, so now let's now move on to the next unit, right? This is only about the rRNA, the RNAs. Now I will give you explanation of the gene. Okay, so what is a gene? Gene concept. When you talk about the gene, what is a gene? Gene actually went on modifying its meaning time to time. But basically, first one what Mendel told, the same thing is gene till today. According to Mendel, so we call it as, now we learn about the gene concept. Gene, according to Mendel, is the unit factor that controls the expression of one character. Till now we call it the same thing as a gene, right? So gene concept is the next unit I am giving you. What is gene concept? Explain how the concept of gene came into existence and how it went on modifying. The first concept was Mendelian concept. Mendelian gene concept. Mendelian concept. According to this, genes are nothing but the unit factors. Unit factors that control the expression of character, and they are having pair of alleles. This is what Mendel explained, right? So according to Mendel, gene, he did not use the word gene, but he said unit factors are those internal characters, internal factors that controls the expression of a character. It may be the color of the flowering pea plant, maybe the height of the pea plant, everything is controlled by this unit factor according to Mendel. It was very long, 18th century, but later the things modified. The word or the gene word are termed by the scientist, second one, that is Johansen. Johansen gave the term gene, and according to him, gene is a physical hereditary unit. It is the physical and structural and functional unit of DNA is called as a gene that controls expression of a character. That was actually gene in the modern way. So in DNA, you must be knowing huge DNA will be there, long one DNA around 2 meters of one DNA length. It has some small discrete units called as genes. These control the expression of a character according to John. He was the one who termed gene, right? According to this, gene is a physical or the structural hereditary unit of DNA, or structural and chemical unit of DNA, functional unit of DNA is gene, right? Later, third one, there was one concept brought by Beadle and Totham. Beadle and Totham. They proposed one theory, namely one gene, one enzyme concept, or one gene, one enzyme hypothesis they prepared. Hypothesis. What is this actually? According to their theory or the hypothesis, one gene is nothing but one enzyme that bring about one physiological reaction, right? Any reaction to take place, an enzyme is required. That enzyme producing unit on DNA can be called as a gene, according to Beadle and Totham, right? According to them, gene is the unit on DNA that codes for a protein that acts as an enzyme for bringing out certain reaction, right? This is called as gene according to Beadle and Totham, right? Now the gene modified, the word gene got modified. Then the last one I'll give you, it is about the modern concept of gene. This you remember properly. Modern gene concept. What is this saying? According to modern gene concept, gene is having not one particular word or function. It has different multiple functions. Based on the function of gene or DNA, we can call it with the different names. For example, gene. This was given by scientist Samuel Benzer. Name of the scientist is very important because the Benzer's gene concept. Okay. According to Benzer, gene is having different names based on its function. First one, we can call gene as cistron. Remember, cistron is nothing but unit for or unit of protein synthesis. Protein synthesis. So, the one that helps in protein synthesis is called as a gene. Means part of DNA that is involved in the formation of proteins, that is giving the information to m-RNA for a particular protein, can be called as gene, and its name is cistron. First you write these upper things, then I will explain details of modern gene concept. Okay, so this you understand. So let me give you this above so that I can draw and explain you. Okay, so let me explain once again modern gene concept by Benzer, because the Benzer's gene concept. Okay. So according to Benzer, so I am telling you about the Benzer's gene concept. We call this as the modern gene concept. Modern gene concept or the modern concept of gene. Okay. According to him, gene can be called as a cistron, which is the unit of protein synthesis. Unit of particular protein formation or protein synthesis. Okay, which part form the protein, that is the unit of protein synthesis. Right? So what is that gene then? Suppose, imagine this is the double stranded DNA. From here to here I am writing the double stranded DNA. It has all the information of many genes, right? Many proteins. Suppose information for one particular gene or one particular protein from here till here it is. This can be called as a cistron. And this is a gene when it is having information for a synthesis of a particular protein. Then gene can be called as, second one, it can be called as a recon also. Recon is unit of recombination. Recombination means crossing over. Means gene need not be so big to bring about one protein, or it need not be so long only. It is called a gene if it has the information for the synthesis of particular protein. But gene also bring about the recombination. Suppose before this, if there may be this much portion is involved in crossing over, this is smaller than this, right? This can be called as a recon. Recon is a unit for recombination. Crossing over happens between the area of the cistron. It may not be whole cistron involved in recombination. Any part can cross over, right? That can be called as gene. Called as a recon, unit for recombination. Third one, it can be called as a muton. Remember the words, whatever I am using. Muton is unit of mutation. What is mutation? Mutation is the sudden heritable change, right? So what is mutation is any change happening in DNA resulting in a change in character or protein is called as a mutation. That becomes hereditary. That is called mutation. So mutation can be still smaller than recon. It may be having very small unit like this portion. This can be called as muton. This can bring about one change in one character becomes hereditary, and it will be as small as single nitrogen base. Nitrogen base also may change and causing change in the character. Like that example I will give you. One of the example is sickle cell anemia, where a single nitrogen base will change, causing the defective hemoglobin and defective RBC. Therefore, muton is the unit of DNA that can bring about mutation. What is mutation? The sudden heritable change is called mutation. And how big is the mutated unit of DNA? It can be as small as one nitrogen base, right? It may be whole gene, it may be one nitrogen base, also bringing about mutation. Therefore, gene can be expressed in three terms. One is cistron, one is recon, one is muton. What is cistron? Cistron is a unit of DNA that can bring about the protein synthesis, or the unit for protein synthesis is called as cistron. Then what is recon? It is a unit of DNA which is normally smaller than the cistron, that can bring about recombination during crossing over. In between, the crossing over happens between the cistrons only. Smaller part may undergo crossing over, exchange the characters, exchange the nitrogen bases. That can cause recombinance. Therefore, it is called the unit for recombination. Also can be gene. Genes anything which bring about a new character, or which bring about a character. Character is not just always protein, it may be recombination also. Then gene can be still smaller than recon. It can be called as muton. Muton is a unit of DNA that bring about mutation. Mutation happens by change in a single nitrogen base also, as it is seen in sickle cell anemia, and some more other diseases are there. They are called mutational disorders. They cause point mutation, where one nitrogen base change also will result in a big defect. That is called sickle cell anemia, where they suffer from defective RBCs, whose RBC becomes sickle shaped. Normal RBCs are this biconcave shaped, but sickle cell RBCs are like this. These cannot carry oxygen. So sickle cell anemia patients will have difficulty in oxygen transportation because they have sickle shaped RBC because of the defective hemoglobin. Hemoglobin is defective because there is change in the nitrogen base, there is a mutation in the nitrogen base. One nitrogen base, GAA becomes GUA, it causes the different amino acid. Instead of glutamic acid, they are forming valine. That is why they will have big problem, that is sickle cell anemia. Therefore, that can bring about change in the character, that can be called as a gene. So, gene can be as big as cistron, as small as muton. That's what is a gene concept which is given by Samuel Benzer. We call the Benzer's gene concept. Understand, Benzer's gene concept is a modern gene concept and we are accepting it. Right? Some of them they write replicon also, but replicon is a whole DNA itself undergoing replication. But these are the three things you have to remember: cistron, ripon and mutant. Okay? So after learning this, I will enter into the gene type of gene in prokaryotes and eukaryotes. Okay? So first you write them down, then we are going to continue with the types, that is types of prokaryotic gene and the type of eukaryotic gene. Let's move on to the gene, right? The gene in prokaryotes and eukaryotes, how they look like. So gene, I told you, it is a structural and functional unit of DNA that controls the expression of a character, right? So genes in prokaryotes are different from eukaryotes. So let me first give you prokaryotic gene. What is a prokaryotic gene? Prokaryotic gene, we call it as let's now talk about the structure of a gene. Okay, how are the gene look like? How they look like in prokaryotes and eukaryotes? So first one, when I talk about the humans and other eukaryotes, genes are called as the monocistronic genes, whereas in prokaryotes we call it as the polycistronic gene. So first one, I will give you prokaryotic gene. Prokaryotic gene. Okay, first I will explain this, then I will go to the eukaryotic gene. So prokaryotes, as I told you, they are called as polycistronic genes. What is polycistronic gene? The gene that produces many cistrons is called as polycistronic gene. How it looks like? Imagine, let me give you the diagram of that. Suppose this is a double-stranded DNA. So I am first drawing DNA, double-stranded DNA. Okay, better always you draw proper DNA which looks like the double helical structure only. This is the double-stranded DNA. This is the structure of DNA. Okay. So what is gene? Suppose from here till here is called as the gene for this. This is gene in prokaryotes from here to here. But it involved number of smaller cistrons because once it undergoes transcription and translation, it can produce more proteins. So it will be having one cistron from here till here. It is called a cistron number 1. Then from here till here, it may be having another cistron, cistron 2. From here till here, it may be cistron number 3. Why it is called so? Remember, whenever this DNA undergo transcription, okay, on transcription I am giving you, if it undergo transcription, m-RNA is formed, right? The m-RNA contain the single structure for all the cistrons, right? From here till here, you get a single m-RNA. Okay, 5 prime to 3 prime. I'm sorry. These are all coding sequences. And in prokaryotes, you don't have anything called as exons and introns, because introns were the internal sequences of DNA that were not coding. But that is true only with the eukaryotes. In prokaryotes, they are not there. All are coding sequences. This is one is for cistron 1. From here to here, cistron 2. Here it is cistron 3. Right? When this undergoes, this is the structure of an mRNA, messenger RNA. Okay? Whenever it undergoes translation, understand this properly. Whenever this undergoes translation, it produces proteins, right? Translation. Based on these coding messages, proteins have to be produced. So the first one, unit 1, the cistron 1 will produce protein number 1. Okay? This forms the first protein, protein 1. Okay? And the second unit, from here till here, it is formed first protein. From here till here, it forms the protein number 2, protein 2. Then from here till here, it forms protein 3. So what do you understand by this? It simply means that one gene is made up of many cistrons that are the informations for many proteins. Therefore, we call it as a polycistronic gene. And it is specially only in prokaryotes. What are prokaryotes? I hope you know. Prokaryotes are those lower organisms that contain not a proper nucleus. That is, nucleus is not covered by the definite nuclear membrane, and they don't have the various membrane bound organelles like mitochondria, chloroplast, all those are absent. Golgi bodies, endoplasmic reticulum, all are absent. But prokaryotes will have this kind of gene which has many cistrons, and when they undergo transcription, they form a long single mRNA for all the cistrons, and all those when undergo translation, they form all different proteins, protein 1, protein 2, protein 3 at the same time. Therefore, we call it as a prokaryotic gene. I want you to draw this diagram and write points on this, right? As you are doing homework, as I explained, based on that you can write the drawing and write points, because each structure, polycistronic or monocistronic gene, is a 2 to 3 mark question. Okay, this you draw and write points on this. Okay, I want you to draw it right now. Okay students, now let us go to the next gene, that is eukaryotic gene. So eukaryotic gene, I have written the diagram as it is, but how it is differing I will tell you. Eukaryotic gene, we call it as a monocistronic gene. Monocistronic gene, that means it is going to form a single cistron. One gene equals one cistron. One cistron is equal to one protein only. That's a meaning. Monocistronic means having single cistron. Means suppose you learn the DNA. DNA is like this, from here till here. Then from here till here, if suppose it is carrying one characteristic, this can be called as a gene, which is nothing but a single cistron. This is whole thing is one cistron only. Means it doesn't have subunits, right? This will have first undergo transcription from m-RNA. m-RNA will have a lot of non-coding sequences in between. These are called as introns. Imagine there is one intron here, there is another intron here, another intron in between. These are all called as a non-coding sequences called as introns. This is another intron. So many introns are there, introns. And what are the coding sequences called? These are called exons. This is called exon. This is exon. So many exons are there. This is the exon. So in between, whichever is a non-coding sequence, that has to be cut, and the coding sequences have to be joined. Therefore, after transcription, once m-RNA is formed, this is called the pro-mRNA. This is not yet ready to undergo translation. This will, after transcription, it will undergo modification. There is something called the purification happening. Purification of mRNA happens, in which all these introns are cut and exons get joined. It is called exonuclease activity. So now what happens, because if the introns are cut and removed, the exons will get joined. This will become a smaller mRNA. This is called the purified mRNA. This is 5' to 3'. It is the purified mRNA. Purified mRNA, what it does not contain? It do not contain any non-coding sequences, because during purification of mRNA, the introns are cut and removed. Introns are removed. Only exons are present. They are getting joined. Now, whenever this undergo translation, after purification, there will be translation. On translation, it is going to form a single protein only, because it is a monocistronic gene. So, single protein is produced. Only protein 1 is formed. Therefore, we call this as the monocistronic gene. So, how to distinguish? They will ask you the distinction between monocistronic and polycistronic gene. First, let us finish the monocistronic gene, then go to the differentiation. So, in eukaryotes, we get monocistronic gene, means one gene equals to single cistrons. One cistron equal to one gene because it is going to form single protein after protein synthesis. Suppose this is the DNA. If this is a gene, from this part to this portion is a gene, it contains the information for only single protein. Therefore, this gene can be equal to one cistron. Therefore, monocistronic gene we call in eukaryotes. Whenever this gene undergoes transcription, forms a long mRNA in which number of non-coding sequences also will be there. This is called the pro-mRNA, which is not ready to undergo translation. Now, in next activity, see all these exons are the coding ones, introns are non-coding sequences. During purification, what happens? mRNA will modify where all the introns are cut and removed. This is called the exonuclease activity. Cutting, removing. Exons, meaningful ones are joined. This is called as a purification of mRNA where introns are cut and removed, exons are joined, forming a meaningful small mRNA. See, it was so big earlier, now it becomes a small with all meaningful nitrogen bases brought from DNA. This mRNA undergo translation, form a single protein, form one protein on translation, therefore it is called as monocistronic gene. Okay, so how to distinguish polycistronic and monocistronic gene? First thing, polycistronic gene is found in prokaryotes, monocistronic gene found in eukaryotes. Okay. Second thing, in the polycistronic gene, there are single gene is made up of many cistrons, more than one cistron. Therefore, the name polycistronic. In monocistronic gene, one gene consists of single cistron. Third one, in prokaryotes or in polycistronic gene, whenever transcription happens, m-RNA will be formed having information for all the cistrons, what they are coding for different proteins, all will be at a time transcribed to form one m-RNA. Whereas in eukaryotes, what happens? Whenever transcription takes place, the whole gene or the cistron undergo transcription to form a single mRNA for single protein. Okay, next. During translation, in polycistronic gene, all the different cistrons that are transcribed to mRNA will be translated to different proteins. Means many proteins are produced at the end of translation. But in eukaryotes, single protein is produced. Before that, one more activity happens. That is, the mRNA which is formed on transcription undergo modification where the non-coding sequences are cut and removed, coding sequences are joined to form a smaller meaningful mRNA or purified mRNA, which is not happening in prokaryotic or the polycistronic gene. Therefore, here the mRNA will have only the nitrogen bases or the coding sequences present together, or they are all called as exon. They are joined to form a smaller one, and it is meaningful. When translation happens, it forms a single protein. There many proteins are formed, and that is characteristic of all the prokaryotes. This is a characteristic of all eukaryotes. Did you understand? So now is your homework. What is that you are doing? You are making two columns. One side polycistronic gene, one side monocistronic gene. Differentiate. But first you have to draw the diagram. So I expect you to draw this diagram, understand, write the notes on this. Then next work is to distinguish between the prokaryotic and eukaryotic gene or polycistronic and monocistronic gene. Do that now, because that question will be asked for 5 marks. Distinguish between polycistronic and the monocistronic gene, or distinguish between prokaryotic gene and the eukaryotic gene. So draw this diagram. Be ready with this drawing, then only you will be able to distinguish between the two genes.